All right, so we have mitosis notes today. You'll need this chart. If you don't have this chart, you can either write it down and get the chart from me the next day, or you could just write it in your notebook without this chart. So we had already talked about interphase. So if you were in class and we already talked about interphase and you have this written down, you don't need to write down the stuff about interphase. But reminder, the cell is not dividing in interphase. So this is not actually part of mitosis. During this time, the cell is going to copy its DNA, it's going to grow, and it's going to copy all of its organelles. Because if we're going to make two cells from one cell, we need copies of everything. This is when the cell is doing its normal everyday job. And if you notice, our DNA looks like squiggles here, and those squiggles are called chromatin. And it's kept in the nucleus. So if you need more time to write this, go ahead, just pause the video. I'm going to move on. Next up, we have prophase. So we're going to go ahead and write prophase here. During prophase, we see chromosomes. So our chromatin organizes into chromosomes. You see it kind of looks like spaghetti here. We have proteins, these little circle balls that help organize it into a chromosome. So proteins are super helpful. Remember, it's the job of the cell to make proteins, but also they make up the cell membrane. They're channels and receptors. Our nuclear membrane dissolves, and that's just so our chromosomes can move around because they're going to have to go half on this side and half on the other side. And then we get strings. So eventually, these little strings called spindle fibers, they're going to attach to the chromosomes, and half of the chromosome, one copy of the chromosome, is going to go this way, and the other copy of the chromosome is going to go this way. Again, pause if you need more time to write here. Here's an example of a cell in prophase. Notice we can see the squiggles. Those are the chromosomes. Okay, this is another example of what a cell in prophase would look like. We don't see any nucleus, right? This guy is in interphase because we see a nucleus there. Next up, we have metaphase. So our abbreviation is PMAT. So P is for prophase, M is for metaphase. Meta and middle both start with the letter M. This is when chromosomes are in the middle of the cell. So chromosomes are going to line up at the center of the cell during metaphase. This is what a cell looks like in metaphase. We have all of the chromosomes lined up in the center. You see there's another one here. PMAT, next up, we did prophase, metaphase. We have A for anaphase. So we're going to write anaphase here. You'll notice our chromosomes are pulling apart to either end of the cell. All right, so remember these chromosomes, they're copies. One copy goes to one side, one copy goes to the other side. Here's an example of a cell in anaphase. We can find a couple more, like here's one, they're pulling apart. Here's one where they're pulling apart. Then we have telophase or telophase, doesn't matter to me how you pronounce it. And our nuclear membrane reforms as kind of a protective barrier to kind of hold those chromosomes in place and protect them. Our cell kind of starts to divide. It's going to pinch in as our nucleus reforms. Here's an example of a cell in telophase. They're kind of hard to pick out. All right, because they kind of almost look like two separate cells. And then last but not least, we have cytokinesis. Cytokinesis is when it fully splits into two cells. And it actually means our cytoplasm is splitting in half and our cell membrane is going to totally split in half as well.